Hello, everyone. We are going to talk about cell division, and I'm doing this in a recording so you can take your time. You can listen to things a couple of times if you need to. You can pause it so you can get everything written down. Just take your time. It's hard to do this over Zoom, so what we would normally do in the classroom sometimes just doesn't work over Zoom. What you do need to have is your paper with the notes. So make sure you've printed that out of Canvas. And once you've got that in front of you, then you can continue with this recording. So before we start talking about cell division, let's talk a little bit about DNA. We're actually going to study this in our next unit. So right now, all you need to know is that it's actually called deoxyribonucleic acid. And that is such a mouthful that we usually just say DNA. Most people don't even know what the full name is. They just know what DNA is. And that contains genetic information. And we'll get deeper into that in our next unit. So the DNA is in the nucleus of the cell where it's protected. And most of the time, it's called chromatin. So this is a picture right here of a nucleus of a cell, and you can't see what we typically call chromosomes. We'll talk about those in a moment. It's just this loose DNA, and it's so thin that you can't see a single piece of DNA. So I know you normally think of DNA looking like this, the double helix, but that is so small that even under our microscopes, we can't possibly see that. So this is chromatin. It's the loose DNA that we normally see in the nucleus of a cell. So DNA that is not condensed into chromosomes is called chromatin. The DNA is chromatin at all times except when the cell undergoes cell division. And so most of the time when you look at a cell, the DNA is loose. And the reason it's loose and called chromatin is because the cell needs access to all the genes on the DNA. And we're going to learn about genes coming up in the next unit. Now, but if the DNA is all tightly packed into chromosomes, the cell can't get to the genes that it needs. So this is chromosomes. This probably looks familiar to you. You've probably seen pictures another time. It's tightly packed DNA. So condensed DNA. It's only found during cell division. I think of it like moving boxes. Like you don't live your life with all your stuff packed into moving boxes. Because then when you need to get to your clothes or your pots and pans and plates, you can't find them. You can't get to them because they're all packed tightly into moving boxes. The only time you have moving boxes is when you're actually moving to a different apartment or a different house. But otherwise, you have your stuff out where you have access to it. Same thing with the cells. Chromosomes are like moving boxes for the cell. And the cell can't get to the genes on this DNA when it's all packed up into chromosomes like this. So the only time you see chromosomes is on moving day when the cell is about to divide. Other than that, the cell's DNA is all chromatin, like in this picture. So the cell has access to all the genes. So this is what a chromosome looks like, and I want you to draw this. There's a space on your notes for you to draw this. Here's the cartoon version, and then here's an electron microscope picture of it. So it's tightly compacted DNA, and there's actually two copies of it. So you can see like this DNA doesn't actually come together. There's one strand of DNA here, and on the other side, there's another strand of DNA, and they are exact copies of each other. We call each side a chromatid. So a chromosome is two chromatids joined together at a centromere. And with human chromosomes, there's always a short arm and then a long arm. And that's one way that we can identify what chromosome it is. Where is the centromere located, and how long is the short arm and the long arm? So two chromatids joined together by a centromere is called a chromosome. And then at the end of the chromosomes, when you look at a real picture of a chromosome, you can't see this. So this is just an artist's rendition of it, of chromosomes. But at the end, there are these things called telomeres. And they're at both ends of both chromatids. And every time a cell divides, the telomeres get a little bit shorter. And finally, after a cell's divided about 60 times, the telomeres are so short that it leads to the cell dying. So it's one way that the cell kind of ages, is as each telomere gets shorter. And so there's a lot of research going on into telomeres right now. Are there things that we can do to make telomeres longer? Because the longer your telomeres are, the longer your cells will live. And then we assume the longer you would live. So there are things you can do to lengthen your telomeres, like people who exercise more. When they look at their telomeres for their chronological age, so how many years they've been alive, their telomeres are long for their chronological age. So just by exercising and eating a healthy diet and not smoking and things like that, you can keep your telomeres longer, and that should lengthen your lifespan as well. But lots of research. You'll hear more about telomeres in the future. So we've got an animation for you to look at, but I will show you these in class. You can take a look at it on your own time, but we will look at it in class together. So DNA, this is the picture of DNA that you've probably seen before, the double helix, way too small for us to see. The DNA wraps around these proteins that are called histones. They're little protein balls. It wraps just less than 2 times around, and those protein balls, the histones, are packed together closely, and they wrap together, and that's how all this DNA is packed together into chromosomes. So, for instance, a human cell, one human cell, all of the DNA would stretch out about 2 meters. That's about 6 feet. And it's so thin you would never see it, and it could break really easily. So that DNA, 6 feet of DNA, is packed around the histones and then packed into chromosomes, 46 chromosomes altogether. And that's how the DNA doesn't get torn apart or lost when a cell is dividing, because it's so packed tightly like this. So we're going to talk in class about homologous chromosomes. So on that part of your notes, just leave that blank and we'll work on this together. So once the cell is, well, that's just some background on chromosomes. So we're going to talk about this cell cycle now. So there's this time that we call interphase. That's actually the longest part of a cell. It's interphase. And the first part, I'll show you an overall view of the cell cycle later. But part of interphase is called gap 1, or sometimes it's shortened to G1 phase. It's right after a cell's divided. So you can think of it as like a little baby cell. Now, what does it need to do? It needs to grow. So inside, there's all these organelles. Remember, we talked about organelles. They each have a job to do in the cell. Well, it needs to make more organelles, like more mitochondria, more ribosomes, and the cell generally gets larger. Then there's a period that's triggered when the cell gets big enough. It can go into what's called synthesis, or the S phase. this is when the DNA is copied. And we're going to talk more about this phase in our next unit, how DNA is copied. So this is a picture of DNA copying, but we'll talk more about that later. For now, you just need to know that the DNA is copied during the synthesis phase. And then it goes through gap 2 or G2. It's just one more growth spurt right before the cell divides. And during this, there's twice as much DNA in the cell because it just copied the DNA in the synthesis phase. Then a period of mitosis starts. Mitosis is only the division of the nucleus. And these are the chromosomes inside a cell. And then they line up in the center. The chromosomes get pulled apart. So one chromatid goes to each side of the cell. And then you end up with 2 nuclei in one cell at the end of mitosis. So you start with one nucleus, and by the end, there's 2 nuclei. And this whole thing in a human cell, it takes about 24 hours for this to happen. So here's what happens during mitosis. The first thing, DNA condenses into chromosomes. So it's like packing those moving boxes. The DNA is already copied, remember, back in synthesis. And now it just gets packed up because the cell's about to divide. So to stop the DNA from getting damaged or lost, it gets packed tightly into these chromosomes for moving day. Then the chromosomes line up in the center of the cell. The next step, the chromosomes split apart, and one chromatid moves to each side of the cell. Remember, each chromatid is an exact copy of each other. So it's going to get pulled, one copy to each side of the cell. And then mitosis ends when the chromosomes are on the opposite side of the cell. So if we go back to the last slide, here's where the DNA condenses into chromosomes, moving day, and then they line up in the center, and each chromosome has got 2 exact copies attached together, 2 chromatids attached together at the centromere. So one chromatid goes to this side, one to this side for every chromosome. In humans, there's 46 chromosomes. So there'll be 46 single pieces of DNA on this side and exact copies of each of them on this side. And then the DNA starts to uncondense and a nuclear membrane forms. And by the end of mitosis, you have 2 nuclei in the same cell. And then one more thing has to happen. The rest of the cell has to divide. So mitosis was just dividing the nucleus. So now cytokinesis. That's the division of the cytoplasm. The cytoplasm is everything outside of the nucleus. So it's liquid and it's lots of different organelles. They just get divided up between the 2 cells. And at the end of mitosis, if everything went right, you've got 2 cells that are identical to each other. And that's the goal. 2 cells that look exactly like each other. And we'll take a look at this website in class together and go over exactly what happens. All right, feel free to watch this as many times as you need to and make sure you've got all the notes down. And we'll go over the videos and the homologous chromosomes in class. All right, I'll see you back in class.